of the metabolic pathways occurring in a cell, with all these white bars here representing specific metabolites. Don't concern yourself with actually reading this. So the type of metabolite or polar metabolites we would encounter in these kind of systems are, for example, amino acids, sugars, polyols, among others. Now, moving on to the definition of the metabolome, it refers to a complete set of metabolites within a biological system. So, again, if we're looking at the scheme of the cell here, we can see that indeed these systems contain more than one metabolite. So if we're talking about the metabolome of the cell, we are simultaneously referring to all of these white bars, or metabolites, and their concentrations within the system. And here comes the really important part. The metabolome is dynamic. So if we were to talk about the metabolome of a human being, we'd find it changes based on various factors, such as your diet, if you're exercising or not, if you're sleeping or awake, if you've been exposed to toxins, if you're taking medication, etc. All this will in one way or another be reflected in your metabolome. That's why we have this entire field dedicated to the analysis of metabolomes in biological systems, because it contains so much information. Now, when it comes to the analysis of these polar metabolites, there have been some instruments which have been around for some time. The first of which is GCMS, which is known for its high efficiency and reproducibility, but requires that our analytes are volatile and thermally stable, and thus a deratization step would be required when we are analyzing more polar compounds. Another technique is LCMS, and more specifically hydrophilic interaction liquid chromatography, and the big plus here is that we don't require a deratization step. And HELIC has actually been quite satisfactory for the analysis of polar metabolites. However, overall observed long analysis times, poor reproducibility, and poor resolution makes us yearn for something that might even be better than HELIC. And this leads us into a technique called enhanced fluidity liquid chromatography, and a series of papers published by the Ulesius research group, with one of the more recent ones cited at the bottom of the slide here. Now, in these papers, they found that EFLC produced narrow peaks, higher resolution, and short analysis time when separating a group of nucleotides compared to a regular HELIC method, thus kind of showing the potential of using this system within metabolomic research. And this kind of forms the basis of my thesis. And the aims of my research is to develop an EFLCMS method for analysis of a wide range of polar metabolites and to compare it with an established GCMS method based on linear range, precision, limit of detection, limit of quantification. Furthermore, when doing my EFLCMS method development, I would like to look at how the EFLCMS settings is impacting the retention of my metabolites. Right, so then we can move into methods, and we can start by looking at the general workflow for the GCMS. So first off, an extraction procedure would be conducted in which polar metabolites would be extracted from INS1 cells treated either high glucose or low glucose conditions. Then, these extracted metabolites would undergo a deratization step, and these derivatized metabolites would be analyzed in a GCMS system run in single ion monitoring mode, abbreviated SIM. This SIM method was developed by basically injecting one metabolite standard at a time in scan mode, and then matching the fragmentation pattern of each eluting peak with an online library for metabolite identification. After this matching, a fragment would be chosen to represent like a unique fingerprint for each one of my metabolites in SIM mode. Now, we can also then take a look at how this GCMS method was validated. So for assessing linearity, a 5-point calibration curve was made for each one of my metabolites, and the chosen concentration range for each one of these calibration curves were based on some previous experiments in which I would try to kind of assess the metabolite concentrations in INS1 cells. The calculated limit of detection and limit of quantification was based on the calibration curves, and when it comes to determining intraday precision, I would calculate the relative standard deviation at the lower end of my calibration curves and the higher end of my calibration curves, and then compare them through a one-way ANOVA. For assessing intraday precision, I would do 3 experiments spread across an entire week, in which I would calculate the relative standard deviation at low metabolite concentrations and high metabolite concentrations, and then compare that also through a one-way ANOVA. And then we can look at some of the obtained results from these GCMS experiments. And on the slide here we see an obtained chromatogram from running an INS1 cell extract with the developed SIM method. And on And we can see here that the method was then able to detect 45 different metabolites. And as an example here, I've marked the peak that corresponds to alanine. Now, how we know this is alanine is based on two criteria. The first of which is this unique fingerprint fragment which I've been talking about, and also on its retention time. So based on these two criteria, we can identify all 45 peaks here. We can also take a look at results obtained from the calibration curves, and it became quite obvious early on that the obtained integrated peaks for each one metabolites would need to undergo some kind of correction procedure through different normalization techniques. The ones I would use is either internal standards, if I had it for the metabolite, scores from a principal component analysis, or in some cases it was just best to leave the data uncorrected because that produced the best linear fit. And on the slide here we see an example of this correction procedure for phenylalanine, and hopefully you can see that correction with principal component analysis yields a slightly better linear fit compared to the uncorrected version. And then through these corrections I was able to obtain good linearity for 35 different metabolites. Now, then through these calibration curves, one can start calculating the metabolite concentration in these INS1 cells treated either high glucose or low glucose conditions. And on the slider we see some representative metabolite concentrations. Now, for all my 35 metabolites that I calculated concentration for, I could see two common trends. The first of which is noted by the metabolites here marked in blue, in which their concentration will be above the calculated limit of quantification. The other group is represented by these metabolites marked here in green, in which their concentrations would fall below the limit of quantification. Now, for the majority of my metabolites, their concentrations would unfortunately fall below the limit of quantification. However, I did state that a lot of them showed good linearity, so one could maybe imagine applying this method to another biological matrix, for example plasma, where you have overall higher metabolite concentrations. That might make this method perform better, and we might yield better quantitative data from that. And then finally we can look at some of the results from the precision calculations, and on the slide here we see the obtained p-values from the one-way ANOVAs. So a p-value below 0.05 represents a significant difference in precision. So for the intraday precision calculations, we can see that for day 1, 2, and 3, there seems to be a significant difference in precision at the lower end of the calibration curve compared to the higher end of the calibration curve. Furthermore, for intraday precision, it seems that the calculated relative standard deviation at lower metabolite concentrations seems to be varying between days, but not at higher concentration. Now, what this all kind of signifies is that calculated precision at lower concentrations seem to be less precise than compared when we're calculating metabolite concentrations or at higher concentrations. And then just to briefly summarize everything that has to do with GCMS, so we were able to detect 45 metabolites with the developed SIM method, showed good linearity for 35 of these metabolites. Majority of metabolites unfortunately had a calculated concentration below the limit of quantification, and there seems to be that we have poor precision when we're calculating at lower metabolite concentrations. And that kind of summarizes everything that has to do with the GCMS part, so we move on now to looking at methods and results for the EFLCMS section. And we can start off by looking at some instrument settings. So when it comes to the chromatography part, I would use a mixture of methanol and buffer as my modifier, and I would use Waters X-Bridge as my column. Now each experiment was basically performed by injecting a mixture of metabolite standards and then operating my MS in positive mode and in scan mode. So when conducting these EFLCMS experiments, I would use a one factor at a time approach when varying all my factors. So basically you have one factor, you vary that factor, and then you keep the other ones fixed, move on to the next factor, vary that one, and so on. The factors that I chose to vary were methanol buffer ratio on the modifier carbon dioxide ratio on the mobile phase, AVPR pressure, column temperature, and flow rate. Each one of these conditions was measured in triplicates, and they were evaluated based on their impact on the metabolite retention factors, and this was done through partial least squares. Right, so then we can look at some of the obtained results from these EFLCMS experiments. And on the slide here we see a representative example of a extracted ion chromatogram showing the 15 metabolites that I was able to detect with my method. Now, how I was able to identify each one of these peaks was basically through injecting one metabolite standard at a time, and then looking at the retention time and the mass to charge ratio value that would correspond to each peak. And through this, I could identify all these 15 metabolites. Now, when it comes to evaluating the relationship between the EFLC-MS settings and these metabolite retention factors, I would do this through a PLS loading plot, which is shown here on the slide. So first off, these blue circles here represent the metabolite retention factors for each one of my 15 detected metabolites. And if we look at the zoom in here of this region here, they seem to be clustering according to their elution order, with valine being a very early eluting metabolite and asparagine being a late eluting metabolite. Furthermore, the green circles here represent the EFLC settings that I varied. So T is temperature, ABPR is the ABPR pressure, U is flow rate, B% is the buffer percentage in the modifier, and M% is the modifier percentage in the mobile phase. So, interpretation-wise, in the context of this figure, we can focus in on modifier percentage and buffer percentage. Now, they are furthest away from the plot's origin, and they are on the opposite side of the plot's origin relative to my retention factors. Now, what that means is in this model, buffer percentage and modifier percentage has the highest impact on retention of my EFLC-MS settings, and they have an inverse relationship to my retention factors. Meaning, of course, if I would increase buffer percentage and modifier, or modifier percentage and mobile phase, we would see a decrease in retention for all my metabolites. I can also note here that temperature, ABPR pressure, and flow rate are clustering close to the plot's origin, and in the context of this plot, it will mean that they don't have a significant impact on retention relative to my other two factors here, and thus I will not focus on these further. The red circle here basically is an added interaction variable, which basically states that there is an interaction between modifier percentage and buffer percentage. And the yellow circles here are added squared variables. Now, they kind of show here that there seems to be a nonlinear relationship between buffer percentage and retention factors and modifier percentage and my retention factors. And what I mean by nonlinear relationship, well, I give you this figure here, in which I plotted some retention factors for certain metabolites against modifier percentage. Now, as we see here, as I'm increasing modifier percentage, retention is decreasing, but is it decreasing so in a nonlinear fashion? And this is following a second-degree polynomial function, I think this is fitted to. Now, one could further explore how buffer percentage and modifier percentage is actually affecting the retention of my metabolites. And remember that I said that the retention factors are clustering according to their elution order of the metabolites. Now, that might serve as an indication that modifier percentage and buffer percentage are affecting the early and late eluting metabolites in a different way. And we can visualize this by plotting some response counterplots, and on the slide here we see this one for valine and asparagine. Now, to just understand the color scheme here, red signifies the area where we have the highest value of our retention factor, and blue signifies the area where we have the lowest value of our retention factor, and then the colors are just what's in between. So if you look along the y-axis here, which represents the change in modifier percentage in the mobile phase, if we compare these two plots, the color scheme doesn't seem all that different. But as we're moving along the x-axis, which signifies the buffer percentage and the modifier, there seems to be a difference here at higher percentages when we're looking at the plot scheme for valine and for asparagine. Now, we could just evaluate this further by just plotting the retention factors either against modifier percentage or buffer percentage for valine and asparagine, which is done here on the slide. So as we see for the slide on the left here, for valine and asparagine, sure, they're decreasing as we are, the retention is decreasing as we're increasing modifier percentage, but they seem to be decreasing in a similar fashion. For buffer percentage, however, the slide on the right here, sure, they're both decreasing, but there seems to be a difference in how they're decreasing. And this can also be evaluated then again through a two-way ANOVA, where we basically look if there's a significant, if there's any statistical significance that there's an interaction between the amino acids and buffer percentage, or the amino acids and modifier percentage. And as we see here for the p-values for amino acid buffer percentage interaction, there is a value below 0.05 signifying a significant interaction, but not so for amino acid modifier interaction. Right, so just to summarize everything which I've talked about up until this point, for GCMS was able to detect and separate 45 polar metabolites in INS1 cells, and 35 of these showed good linearity. But, as I mentioned, a lot of the metabolites had a calculated concentration below the limit of quantification, which might question the validity of using this method on INS1 cells, but if we were to apply this to other biological matrices, maybe we would obtain better quantitative data. As for the EFLCMS part, methanol buffer ratio and modifier carbon dioxide ratio seems to have the largest impact on retention, and it seems that also the buffer ratio, or the methanol buffer ratio, seems to be impacting early and late eluting metabolites differently. And as for the future aspects here, so as it stands right now, the EFLCMS method is quite at an early stage in its development. This has been mainly due to some technical difficulties with mass spectrometer and also due to a lot of time constraints. But if I were to maybe move forward with this project, the first thing I would try to assess is, could I detect more metabolites with my EFLCMS method? And I would try to do this by maybe operating my mass spectrometer negative mode, or I would change the mass spectrometer altogether because I've been experiencing some technical difficulties with the QDA in the past. It would be also interesting to look at more into how the buffer may be impacting chromatography in this system by maybe varying the salt type, salt concentration, and pH. Also, one could maybe look into different collant chemistries and modifier types as well. And then also another thing would be maybe to look into the uses of UHPLC columns. So with the system I've been using right now, there's a technical limitation when it comes to the maximum system pressure that it can handle. If one were to work with more state-of-the-art systems, where you could have a higher maximum system pressure, you could decrease the particle size also, and maybe you could then start working with UHPLC columns. And then finally, of course, a proper EFLCMS method optimization and validation will be required here for me to even compare it to my GCMS method. And also within that, it's also implied that I will need to also develop a sim method for my MS, for the MS part in the EFLCMS. Right, and then finally, just some acknowledgements. I'd like to thank Peter, Claudia, Sofia, Katarina, and everybody in Irene's group in GTG. It's been a long and bumpy road, but finally I'm at the end here. So thank you all for your support, and thank you for listening.
Thank you. Okay, so questions on this?